Welcome. Thank you for joining the Eastern Brook Trout Joint Venture Swimming with Science series. My name is Dave Kazyak, and I'm a research fisheries biologist at the U.S. Geological Survey Eastern Ecological Science Center, where I lead a conservation and genetics and genomics laboratory. Our team works with stakeholders from across the eastern U.S. to support the conservation of a variety of rare, threatened, and endangered species. But for today, I want to focus on brook trout. Our team's been working on brook trout for roughly 20 years, and to date we've looked at the genetics of roughly 25,000 individual fish from about 800 populations. And that body of work has really opened my eyes to some of the challenges facing our small, isolated populations. So for today, I want to talk about the importance of isolation, drift, and genetics for the conservation of wild brook trout. So let's talk about brook trout conservation. For most people, the primary way to interact with wild brook trout is through fishing for them, and anglers have really helped to catalyze many management and conservation efforts. And if someone wants to go fishing for brook trout, they're likely going to want to know things like, where do brook trout occur? Are there enough trout there to provide a rewarding experience? Or how large are the fish? And to a significant extent, brook trout management traditionally has emphasized those demographic characteristics in order to provide opportunities to anglers. Through the efforts of agencies, universities, and NGOs, we've collectively generated a tremendous amount of data on the abundance and distribution of brook trout throughout their native range. And through those demographic surveys, we know that many of our historic populations have become extirpated, and a significant proportion of those that remain are in a diminished status, typically fewer fish which are restricted to a smaller habitat patch. This figure from Mark Hoodie and others seems to show up in so many brook trout presentations, to the point where it almost feels requisite, and I think that's because it's alarming and highlights the extent to which brook trout populations have contracted and disappeared across the eastern U.S. In the years since that map was published, a huge body of research has explored various factors which impact where brook trout occur, their densities, and their size structure. And generally speaking, larger, intact forested watersheds that can provide access to diverse habitats are favorable from a demographic perspective. However, surveys have also highlighted the extent to which brook trout are often isolated in small headwater habitats. In some cases, these populations are isolated by natural barriers that prevent passage and gene flow. Waterfalls are perhaps the most obvious, but other natural barriers, such as areas of unsuitable habitat, can also create impermeable barriers to fish passage. It's not always the big flashy ones. Throughout the native range of brook trout, there are many populations above waterfalls and other natural barriers which have likely been there for a long time. And some of those populations are apparently fairly stable. These naturally isolated populations may be, at least to some extent, adapted to the conditions. For example, don't swim over the waterfall, as you'll have a one-way ticket to exit the local gene pool. There are also some aspects of brook trout ecology which likely help them persist in isolated habitats. Things like phenotypic plasticity and life history variation. Now, having acknowledged natural barriers which isolate populations, we also know that humans have radically altered the landscape and dramatically fragmented habitats over the last several hundred years. Dams generally get most attention in the context of fish passage, but for brook trout, there are many other barriers that we need to be concerned about. Culverts often have poor fish passage, and those issues are generally not as well documented on the small headwaters where so many of our brook trout populations occur. And this problem of fragmentation is quite pervasive. Currently showing a map of stream passage barriers in catchments that were predicted to support trout in a modeling study by DeWeber and Wagner. In this case, I think the model considered any species of trout, not just native brook trout, but within the Chesapeake Bay watershed alone, there are over 5,500 barriers within trout catchments, of which 40% were rated as at least a moderate barrier. We also know that invasive or introduced species such as brown and rainbow trout are associated with reduced gene flow in brook trout, so there can also be biological barriers. Unsuitably warm stream temperatures such as those found in developed areas and acid mine drainage both present obstacles to connectivity for brook trout. Overall, I think the maps I showed a moment ago are likely to be a significant underestimate on total passage barriers, since they don't include barriers like acid mine drainage, unsuitable habitat, or invasive species. And if we look across the eastern landscape, we can see quite clearly that most of the patches which brook trout occupy are really quite small. On this slide, I'm showing a histogram of patch sizes based on 2015 data from the Eastern Brook Trout joint venture. The vast majority have a total patch area of less than 100 square kilometers, and roughly half of all documented brook trout population patches have drainage areas of less than 7 square kilometers. In most cases, this translates to limited habitat availability and small populations which are isolated in headwater streams. Another thing to keep in mind is that as some populations become either isolated above barriers or extirpated, connectivity becomes increasingly challenging. We know that we've lost a lot of populations in many parts of the range, and the remaining populations are often more separated from one another than in the past. Connectivity and gene flow, though, are really network issues. So when you lose one population, the network has less connectivity. And if we look back in time, prior to widespread human impacts, it's likely that brook trout metapopulations, and by that I mean systems where gene flow could occur among nearby populations, were more prevalent, as there were likely far fewer barriers to connectivity on the landscape, and brook trout occupancy was greater. But as we've lost some populations and created all these barriers across the landscape, there are now far fewer opportunities for gene flow. And I want to emphasize here that most of these anthropogenic changes are recent, typically within the last 100 years or so. Taken in concert, we now have countless small isolated populations of wild brook trout. These populations are especially prevalent in the southeast, but they're also quite common at higher latitudes. So, having hopefully established that many wild brook trout populations are small and isolated, I want to pivot and talk about some of the special considerations for managing small and isolated populations. In other words, what we might call the small population problem. To start off, small isolated populations are likely more vulnerable from a demographic perspective. We know that many stream-dwelling brook trout populations are highly variable from year to year, with relatively high mortality rates that are offset by pulses of recruitment. Those pulses of recruitment are often associated with stream conditions that can vary a lot from year to year. And overall, small populations are just much more likely to blink out through chance events, like a series of poor recruitment years. In addition, small isolated brook trout populations typically have access to a lesser diversity of habitats, and they might not be able to disperse when conditions become unsuitable, making them more vulnerable to localized disturbance. In general, these populations are just more vulnerable to random demographic and environmental fluctuations. But in conjunction with those demographic concerns, there are also important genetic considerations for isolated populations, particularly for small isolated populations. We know that gene frequencies shift randomly from one generation to the next, and we call that genetic drift. Genetic drift is ubiquitous in all populations, and it basically reflects that inheritance is a finite random sample of genes from one generation to the next generation. Now, in a population that's effectively large, shifts from one generation to the next are very small, essentially because the parent's generation is being sampled many times over to create a new cohort, and the frequencies don't change that much. But in populations that are effectively smaller, the rate of random change increases exponentially, and that can have major implications for conservation. Random change in gene frequencies leads to a loss of alleles and a loss of heterozygosity. In other words, a loss of diversity. Rare alleles are lost faster than common ones, and to make matters worse, these random fluxes in small isolated populations mean that natural selection, the process by which continued adaptation occurs, doesn't work as well in these populations. And all of that is problematic in the context of evolutionary potential. Genetic drift is also associated with inbreeding and can increase the frequency of harmful mutations, both which can be detrimental to fitness. In the previous slide, you may have noticed that I referred to the effective size of populations. Effective population size, which is often abbreviated with an N subscript E, is different than census population size, and it's an often misunderstood term that geneticists use to describe the rate of genetic drift within populations. There are a lot of factors that contribute to effective population size, but you should be aware that effective population sizes are often much lower than census population size in wild brook trout. To illustrate losses of diversity over time in small populations without connectivity, I've plotted heterozygosity, which is one metric that we use to measure diversity using an idealized model for isolated populations. I've started the simulations using the average level of heterozygosity we've seen across hundreds of wild brook trout collections that we've studied. We have generations going forward in time from 0 to 30 on the x-axis, and heterozygosity as a measure of diversity on the y-axis. And what we see is that if we have an effective population size in the hundreds, the rate of loss is very slow. But you can see that over the course of 30 generations, which corresponds to roughly 100 years for many stream-dwelling brook trout populations in the eastern US, it's possible for populations to lose a lot of diversity when their effective population sizes are small and there's no gene flow with other populations. And if we look at those curves for a simulated population with an effective size of less than 25, the effect is really pretty dramatic. Unfortunately, many of our isolated brook trout populations have effective sizes of less than 50 and often much less than 50. So we should expect rapid losses of diversity in relatively short time frames. One thing that this suggests is that isolated populations which outwardly may appear stable may be losing genetic diversity and becoming increasingly vulnerable to extirpation. In contrast, where we have networks of populations with connectivity, we expect to see gene flow which limits genetic drift. So that was a theoretical model, but what do we see when we look across the landscape? Using some populations from Maryland to illustrate, we generally see a clear relationship between genetic diversity within populations and their effective population size. Populations which have larger effective sizes tend to have higher levels of diversity, and as we would expect, our populations with very small effective sizes typically have dramatically less diversity. But when watersheds allow for connectivity between populations, such as in the Upper Savage River watershed, which is shown here in yellow, even small effective populations are more likely to maintain diversity through gene flow. Next, I'd like to shift gears a little bit and talk about the importance of genetic diversity for long-term persistence. One concern is that as populations lose diversity, individuals within a population share more and more of the same genes. Small populations are also likely to have some really messy pedigrees. And as a result, we expect to see increases in homozygosity and potential for inbreeding depression. In other words, fitness consequences. I will say that I'm not aware of much literature that has specifically measured inbreeding depression in wild brook trout populations, but there's strong experimental evidence of inbreeding depression in endangered cutthroat trout, and I've seen some suggestions of inbreeding depression in at least some populations of wild brook trout. But beyond the risks of inbreeding, I think we need to consider the evolutionary future of brook trout populations and what we know about rapid adaptation to change. At this point, it's very well established that our world is changing. It's changing quickly, and it's changing in ways that often have no historical precedent. And those changes are in many cases having sizable impacts on wild trout populations. We know that brook trout are a cold water obligate fish, and they have a broad array of adaptations to local conditions, or rather more accurately, what those conditions have been in the past. Today, many of our trout populations are found in disjunct headwaters with little opportunity for gene flow or range shifts in response to climate change. And already in the last 150 years, we've seen dramatic increases in average temperatures around the world, and we're now on a trajectory to enter the warmest period in more than 100,000 years. North America is projected to get at least several degrees warmer. How much warmer really depends on decisions that are made in the coming years, but the consensus is that we're already locked into a significant amount of additional warming beyond where we're at now. In addition, the projections suggest that extreme heat events will become both more frequent and also more intense. For wild brook trout populations, many of which already face stressful summer conditions, that's clearly cause for concern. We will likely see a little bit more precipitation across the native range of brook trout, which could be a good thing, but the timing and intensity may change from what we're used to, and in turn, what brook trout populations have adapted to. In general, heavy precipitation events and drought are expected to become both more frequent and also more intense. So even in the absence of other changes with things like land use or invasive species, the projected climate shifts are likely to challenge the persistence of many populations and significantly change the selective pressures they face. Clearly, warming temperatures that approach thermal tolerance limits are going to impart a strong selective pressure, but we'll also likely see shifts in the selective pressures on many other traits, perhaps things like spawning phenology. We know that most mortality in wild trout occurs in early life stages, and that flow patterns and ice scour during those periods have a big impact on recruitment. So it follows that selective pressures on spawn timing might shift a lot. Things that have worked well for brook trout in the past might not work as well in the future. So how can brook trout respond to changing conditions? Phenotypic plasticity is one way that trout can adjust to changing conditions, where individuals with the same genetic code express different phenotypes depending on environmental conditions. Phenotypic plasticity is well documented in trout, and it's undoubtedly important when we're talking about rapid changes in the environment. However, since we're also expecting selection pressures to change, genetic changes can be an important pathway to promote resilience. The brook trout genome contains several billion bases of genetic code that provide the instruction manual for the fish and how it's going to respond to its environment. Some parts of that code are used all the time, some are used only under specific circumstances, and still other sections may be relics from the past. But this code isn't just dictating how an individual responds to its current environment, but also how its progeny are going to respond to future environments and their capacity to adapt to change. The rate of change really depends on some factors that are specific to a population. But it's important to note that adaptation isn't always a slow process, and it can actually occur quite rapidly under certain conditions. First, we need to have changes in selective pressures, which are expected given the projected changes in the environment. We need to have phenotypic diversity within a population that those selective pressures can act on, and those phenotypes need to be heritable, based on underlying genetic variation that already exists within the population. In other words, standing genetic diversity. Rapid adaptation is really dependent on standing genetic diversity, which is one of the reasons it's so important to conserve genetic diversity in the first place. Genetic mutation is far too slow to help us out in the context of rapid change. Finally, we know that populations need to be large enough to allow for selection to act efficiently. In populations that are effectively small, random changes from one generation to the next can overwhelm selective pressures. Bearing in mind those factors which provide populations the best chance at rapid adaptation, there are potentially some things that we can do to help promote resilient populations. We can attempt to increase our effective population sizes, which should help maintain diversity and improve the ability of populations to respond to changing conditions. This could be accomplished by improving habitat quality or reconnecting habitats. Restoring gene flow by improving fish passage and reintroducing extirpated populations can also help maintain diversity. These are often actions that managers and conservation practitioners are already implementing, but they're expensive, they take time, and opportunities are sometimes limited by resources or access. And I think all of that underscores the virtues of protecting our existing interconnected strongholds where they occur. But given that our strongholds are relatively scarce in a lot of areas, is there anything else that we might be able to do to support small and isolated populations, many of which seem to already have lost a lot of their diversity? And one potential tool which is being discussed and explored is genetic rescue. And what genetic rescue basically entails is moving a small number of animals from one population to another with the intent of artificially creating gene flow between isolated populations. The goal of genetic rescue is to increase fitness and/or adaptive capacity through the introduction of new genetic material. I personally think this tool has a lot of potential to mitigate many of the challenges we've been discussing, but there are very few documented case studies of genetic rescue in fishes, and I think we would really benefit from some additional experimental tests to see how well it works and in what situations. I'll note that Shannon and I have a paper that's currently in review where we're attempting to synthesize the state of the science for genetic rescue as it applies to brook trout, so stay tuned for that. And as I wrap up, I want to share what I think are some key outstanding questions where we don't currently have enough information. We don't really know the extent to which inbreeding is impacting our small, isolated populations. Seems likely, but it's not something that's very easy to document. We don't really know how much capacity brook trout populations have to adapt to the challenges they're likely to face in the future. While rapid adaptation can occur quickly under the right circumstances, there are still limits, and we don't currently know what those are. As for genetic rescue, we could benefit from more experimental tests to evaluate utility, refine methods, and help zero in on those populations which are most likely to benefit. And finally, there's a paradox that I alluded to early in my talk, in that we also have many populations above barriers, natural barriers that is, that appear to have persisted for a long time. What has allowed those populations to survive? They've had a long time to adapt in situ and potentially purge harmful alleles. But we also don't know how many other populations were similarly isolated and have since blinked out. I would also argue that we should be cautious to assume that those populations have really been there for millennia, as it's possible that some of those above natural barriers were established by people at some point in the past to create a food resource. Finally, given the pace of change, these are questions that I would argue we need to sort out through experimental work as soon as possible so that we're in the best possible position to promote resilience in the future. I want to thank you all for joining the Eastern Brook Trout Joint Venture Swimming with Science seminar series. I hope you'll consider joining our follow-up discussion on Monday, February 6th at noon.